Hi, good day, everybody. Subra here. I work for the DAF. Just a bit of touching the fall armyworm management, focusing on the vegetable crops today. So just give a bit of overview how it can be spread over the last 13, 14 months. It was first detected in January 2012, but within the 13, 14 months, it's gone up to Tasmania, even recently North Fork Islands. Pretty much within 3 months, we could able to see in the Bowen Burdekin area from the early detection. Then they started to establish pretty much into, but the moth can migrate pretty fast. It's pretty much few hundred kilometers each moth can fly. just just give you an idea. We're doing a bit of last year we did some monitoring with using the sex pheromone traps. These sex pheromone attract the male moths into the bucket trap, and we're looking at mainly it's attract fall armyworm, but also it gets some bycatchers called false armyworm and few other ones. The data we got for last year, see that blue line showing the fall armyworm numbers. The early detection was in somewhere in mid-March. So the first 2-3 months, the numbers are a bit low. Then after May, June, the numbers started to increase in a pretty large numbers. So you could see some, even through the winter time, they started more active, then started breeding, and the local population might be the reason you see the numbers keep on increasing. By September, October, the numbers really gone up to like each 2 traps in a farm got over close to 800, 900 moth in a week period. Then when the season get close to the sweet corn season finish, the numbers started a bit drop down, but we could able to see more numbers during the summertime also. So this actually not showing any force period or even the winter time, they can keep on breeding and the numbers are still active. So this giving a bit of idea in a non-corn growing area, like the Gamlu area, there's no sweet corn there. But last year we find only few numbers in the trap. It's only like a under 10 in average. But this year actually the numbers gone very high. See that in May, June, July this year, the same period, we able to get up to over 200 moth in a trap per a week. So then it started to be declining now. So that means, they slowly adapting to the new locations. So this give you a bit of biology of this pest. We did a bit of sampling later part of last year, pretty in the hotter months, October, November, that giving how that the fall armyworm is breeding from egg to neonate larvae, just take only 3 days to catch and produce. So within 3 days, they laid, you see the batch of larvae here, they 100 of them, they're crawling and feeding on that one. They can cannibalize on each other, but still quite a bit of numbers get established. Then from eggs to come to the second install larvae take only 9 days, and eggs to the fourth, fifth, and the larger one within 2 weeks, they pretty much come to that big stage around here. They are the more vigorous feeding sizes. Then they pupate into the soil. Most of them pupate in the soil. There's a few we seen into the fruit or in the copse. But within 3 weeks from eggs to they pupating, they can overlapping few generation in a year. So this give a bit of idea about the host got affected. Most of sweet corn and maize corn are worstly affected crop. That's a more very favorable crops. And also we some moderate low infestation we noticed in sorghum, and this year a bit more on capsicum. Pretty much this strain they everybody predicted is going to be mainly on the grassy type crop, but we seeing more in the capsicum also in some other occasionally we seen on other crops, but didn't cause much damage yet. So this a bit of damage pattern you see in sweet corn. So the early seedlings are more highly sensitive. They pretty much can kill the whole seedling within a week. Then when they go into the vegetative stage, is showing a lot of windowing pattern. Then the tassels, you see the tassels pretty much eaten and affecting the pollination part. And the silk also they damage. And unlike Heliothis, the damage pretty much going the whole copse on one side, or maybe the whole cop got rejected. So the Heliothis mostly we see on the tip of the copse, but this one pretty much goes in the side up to the bottom of the crops. So this bit on a capsicum one, somebody would have seen quite a lot, and Marco experienced quite a lot last year, and he gave me some sample to identification. So this the leaves got a lot of eggs on it. Is the egg masses laid, and we bit hard to see in the early stage, and mostly see them they straight away burrow into the fruit, and they started feeding inside. There's bit not in the sweet corn you could see them very early, but in the capsicum it's a bit hard to detect in the early stage. Even when the fruit get mature, only you see the damage. There's a bit of chemicals, pretty much you people already touched. We got a permit to use on Collagen and Success Neo, Procloim, then the Intoxicarp, Avatar, and Prodigy. These products got rejected, but a few of the older ones like Medomil and Pyrethroid and Chlorophyrifos, the bit of very question mark. Actually, we're not finding any good evidence they are working, and also there's some sample test that we find a high level of resistance for this the carbamate and synthetic pyrethroid groups. And also we working bit of seed treatment and soil application method to see to protect the early part of the crop, and also a few biological stuff. I'll have a bit of talk on that, and some like a luran kilting, attracting the male moth, and then the QMFAW is one of the product has been got permitted to use now or registered to use. So this is a bit of a few last season result. We applied some soil product, two product in the soil at 2 different rates, and also a bit of soil drench and seed treatments. And the control, you see the control plot pretty much had around over 80% of the plant got killed within 2 weeks. So the soil treatment and seed treatment giving 1 to 2 weeks of protection. Then after that, they started to break down. So at least some level of control for early stage, but still not registered this product yet, but it's in the pipeline. And also we got some bit of work as a foliar application with the Collagen at 2 rates. And first one is the label rate, other one is a bit higher than the label rate. Both pretty much work on the similar way. And also the couple of new product, DC1X3 and B101, that's a bit more promising. And the pyrethroid and mixed with pipenol butoxide didn't give much control. Actually, pyrethroid not working, and control pretty much, the scale, the foliage damage score up to 9 is pretty much very bad damage. This one's another virus product called Collagen. Then we done some work with the Collagen to test on the early stage of fall armyworm larvae, like neonate larvae and second instars, 3 and 5 days and 7 days after application. What we did actually, we spray the crop and bring back the leaf material and feeding in the lab to see what level of mortality is giving. The neonate larvae started to die with the Collagen 3 to 5 days time, but the large second instars larvae is taking up to 7 days to give a reasonably good mortality up to full mortality. But it's working, but it's with a slow working product. But we didn't have any good data for the other later stage one. But some information we found in the field saying the larger size are pretty hard to kill with Collagen. So there's a few beneficials we've started to finding in the infested field. One of them, the good one is called spine shield bug. The adult on that side, the top 2 are adult, and the nymph stages and the egg stages. The nymphs are pretty much very aggressive feeders. They can feed around 4-5 fall armyworm small larvae in a day. And the adult also, the females are more aggressive feeders. They feed more, probably 5-6 larvae in a day. They're more like a common predators up here. They are more polyphagous predators. They can feed on most of the soft body larvae. And also there's another one called the white banded shield one, but it's not very common. It's been seen only a few times. But also some earwigs and Assasian bug also we noticed in feeding on fall armyworm. Some ants also, the black ants also taking some fall armyworm larvae. So they probably the bit of inactive larvae is taken out by the ants. This is another important one. We recently find some Costasium parasitoids. They actually come and lay the eggs into the fall armyworm larvae. Then they breed inside and the pupae coming out from the armyworm larvae. And this only last year we didn't see much, but this year we started to see much of the. But from each larvae point, finally we're finding around 15, 20 cocoons coming out. So there's a bit of thing, and in the Collagen and Collagen sprayed flood and Collagen success flood, both actually didn't see much effect on the emergence. So the pupae collected from that field and we rear them in the lab, so pretty much it's a good emergence happening. So seem to be that product seem to be compatible with this parasitoid. Okay, yep. These are the parasitoids, and we're finding like a technid flies. They come and lay the eggs on the armyworm. See that more than around 7-8 eggs has been laid on the back of the one. They pupate inside the armyworm larvae and the flies coming out. Then say another egg larval parasitoid, and I'll talk a bit more later to run out of time. So there's other 2 larval parasitoid also we're finding in smaller numbers. So just in a summary, this is the armyworm moth activity is low during the March, April time last year. Then they started going through the November period, and June to November numbers are pretty high. And the seedlings are more sensitive and need to be protected early vegetative stage, especially for the corn and one. Then success neo-collagens are still more effective ones for younger larvae, and the biological control is a bit more promising now. The Costasium is one of the species we're working on. So that's pretty much. So just to acknowledge the funding one, Department of DAF, Department of Agriculture and Hort Innovation, and the people supporting for the research. Thank you.